Hello, I'd like to welcome you to the CMI webinar series designed for CMI and by CMI. I'm Cynthia Howell, and I am going to be your moderator for today. I want to tell you a little bit about our webinar. For those of you that may not have been on one before, they are hosted by Colorado School of Mines, but broadcasted from many different states at the same time. If you have a suggestion for a future webinar, we'd love to hear about it. Please make sure that you put it at the end of the survey, and/or there's a email right there that you can actually send it to as well. This is a public webinar, and I want to remind you that a recording is going to be available next week at aimslab.gov/CMI. And you can also find it under the webinar menu and on YouTube and the Critical Materials Innovation Hub channel. I want to encourage you also to subscribe to our YouTube channel, because we're putting up a lot of different YouTube snippets that will be of value to you in your research world. Questions, we're going to hold the questions till the end of the presentation. And I would encourage you, however, to make sure that you put those questions as soon as you have them in the question and answer box, so that they're queued up and that our presenter will be ready to take them as soon as she's finished her presentation. I'd like to introduce our presenter today. This is Dr. Alexa Schmitz. She is the CEO and co-founder of REEgen, a rare earth manufacturing startup company in Ithaca, New York. Her PhD was in plant pathology and plant microbiology from Cornell University. As a postdoc in a Barstow lab, she also developed the technology for efficient recovery of rare earth elements using engineered microbes. And you're going to hear a bit about that today as well. Shortly after co-founding REEgen in 2022, was awarded the Activate Fellowship, which has helped support and the early implementation of this startup. But I don't want to take too much more time. I'd like to turn it over to Alexa for her presentation.
Thank you so much, Cynthia. And also a huge thanks to CMI for inviting me here to present to you today. It's really great to be able to share more about what we're doing, specifically what we're doing to build clean, sustainable supply chain for domestic rare earth elements. So as Cynthia mentioned, I'm co-founder and CEO of REEgen. We're a startup based in Ithaca, New York. We're up in upstate New York in the Finger Lakes region. And we are using synthetic biology to build a clean, sustainable process for manufacturing rare earth elements. So I'll just get started with a bit of background. So why are we doing this? Well, we all know that climate change is a huge issue. And what's really remarkable is that with the technologies we have available today, electrification of the huge impacting industrial processes could actually eliminate 50% of their greenhouse gas emissions. But for this to happen, we will need far more critical materials, especially rare earth elements, than we are producing with our current mining capabilities. Rare earth elements, to be specific, are a group of periodic table elements, including the lanthanide series, yttrium and scandium. They all have really special properties, optic, catalytic, magnetic, that make them essential for most of today's electronics. However, they are all individually important, so we really need to separate them into their individual elements, which is often very difficult for these applications. To give you an idea, rare earth elements basically run the modern world. They're in your computer screens, they're in your iPhones, they're in your other mobile phones and high efficiency lighting. Speaking of iPhones, if you want an idea of where they are there, we have magnets in the vibrator, magnets for the speaker. Your color screen has rare earth elements, and even rare earths are used to polish the glass. But really importantly, to combat climate change, rare earth elements are absolutely critical for renewable energy infrastructure, especially those strong lightweight magnets that power wind turbines and EV motors. To give you a sense, rare earth neodymium, the element neodymium, is one of the elements in permanent magnets that really enables much more efficient energy generation. It's just a small part of the magnet material, but if you look at the sheer size of the magnets that are required for each wind turbine, this really adds up. And so a single turbine ends up requiring high amounts of neodymium. So here's the thing. rare earth elements, despite their name, aren't actually rare. They're just very dispersed in the Earth's crust and not really found in concentrated deposits. This makes it really difficult to mine them and refine them, and the conventional processes that are used to do this can be incredibly energy intensive and environmentally damaging. So one of those single offshore wind turbines requires over a ton of the rare earth neodymium. And to get that results in huge amounts of toxic waste. We're talking 65 tons equivalent of CO2. Over a ton of radioactive waste comes out of the mining of thorium and uranium bearing minerals. Because of all of this, the world has basically outsourced the production over time. And actually now China alone is producing over 90% of our global rare earth refined materials. This monopoly is a serious problem. It's causing supply chain risks and even concerns for national security. To clarify what I mean by 90%, here's a breakdown of the rare earth supply chain. So the US is producing rare earth concentrates. MP Materials has reopened the Mountain Pass mine out in California, but the majority of that is still sent to China for separation and refining. So that's what I mean when China is controlling 90%. This is leaving a huge gap in the supply chain, especially domestically and outside of China, especially for the production of rare earth oxides and the reduction to metals to make magnets for renewable energy infrastructure. So at REEgen, just to give you a sense of what we're doing, we are targeting specifically the rare earth concentration step and also separations with biological processes. So to make this happen, we're developing clean biological processes for rare earth manufacturing that can eliminate the high costs associated with those environmental impacts by eliminating the environmental impacts. Also, for our process, we're getting rare earth feedstocks from other materials, secondary materials. So instead of generating hazardous waste with mining, we're actually utilizing waste from other industries to recover these rare earths. This also allows us to unlock billions of tons of new feedstock without additional mining. Then we have genetically optimized bacteria that can leach and purify the metals with greatly reduced environmental impact. Furthermore, along with manufacturing sustainable rare earths, we are converting many of our byproducts into supplementary cementitious materials, working with some collaborators to help make this happen. And this will allow us to bypass landfilling completely and also help offset the emissions of the concrete industry. Overall, our process involves two key biological processes: rare earth bioleaching and bioseparations. For bioleaching, genetically optimized microbes are converting glucose into a leaching solution called a biolixiviant. This biolixiviant, so biological lixiviant, is used to leach metals from a solid rare earth feedstock. That solution of leached metals is then separated into individual rare earths using genetically optimized microbes that selectively bind the different metals and help separate them. So in rare earth bioleaching, at REEgen, we're using the bacteria Gluconobacter oxidans. This converts glucose primarily into gluconic acid and other important molecules. So not only is Gluconobacter one of the best rare earth bioleachers, but it's non-pathogenic. It's generally regarded as safe, which is what GRAS is. And most importantly, it can be genetically engineered, which allows us to improve its performance. The biolixiviant that is produced is then mixed with the rare earth feedstock, solid feedstock, where it leaches the metals into solution through acid leaching and chelation. The rare earth bioleaching with Gluconobacter was actually first demonstrated at the Idaho National Lab, led by a team led by David Reed. As part of this work, they compared rare earth bioleaching with leaching efficiency using a variety of concentrations of abionic gluconic acid. And what they found was that the bacterial lixiviant was still able to leach the metals with higher efficiency than a 90 millimolar solution of gluconic acid, which is actually 6 times higher than what was found in the biolixiviant. So these results really indicate that the bioleaching efficiency really cannot be explained by gluconic acid alone, and that other factors must be playing an important role here. Furthermore, the Idaho team took this research a step further and published a few techno-economic analyses to estimate the commercial potential for rare earth bioleaching, specifically from spent fluid catalytic cracking catalysts, which are used for petroleum refining. This work demonstrated some potential for a small margin of profit, which was inspiring, but maybe not enough to get a huge commercial uptake. Around this time, I was beginning my postdoctoral studies with Buzz Barstow at Cornell University, and we set out to see if we could improve on these numbers with genetic engineering of the bioleaching bug. So the first step for improving the efficiency of rare earth bioleaching was to identify the key genetic factors that underlie this important mechanism. This work was luckily funded by an ARPA-E grant to Buzz's lab. So to do this, we had to first build a whole genome knockout collection in the bioleaching microbe gluconobacter oxidans. I won't go into the details of how we did it. It was rather complicated. You can find it down in this publication here from 2021. I will say it took a lot of perseverance and even some sweat and some tears, but we did it. And once the collection was built, we screened it for strains that had modified media acidification with glucose. We use colorimetric dyes to identify the strains that had modified acidification. And as you can see in this example here, the purple color in wells F7 and G7 indicate that the strain in those wells produced a bioexcipient with a lower pH than the majority of the plate, indicating that the gene knocked out in those strains plays a role in inhibiting bioexcipient production. So when you have it, you inhibit production. When you knock it out, you get better production of bioexcipient. So finally, once we screened all of the knockout strains, there was a strain for each gene in the gluconobacter genome. We were able to identify which ones underlie bioleaching, and then we demonstrated the effects directly on bioleaching of a subset of the strongest hits from that collection. Overall, we were able to identify 165 genes in the gluconobacter genome that influence production of acidic bioexcipient. Most of the knockout strains that we identified had just a modest reduction in acidification when they were deleted. But interestingly, a handful, like the ones in the plate from the previous slide, produced a more acidic bioexcipient. Also, several strains, these here, all but lost the ability to convert glucose into an acidic bioexcipient. When we selected the top and bottom strains from that chart for actual direct bioleaching of rare earth elements, we found that among those with greater acidification, the knockout strains, several actually did have significantly improved rare earth bioleaching. What really stood out, though, is that 4 of these strains had disruptions in the same biological system. This was the phosphate-specific transport system. So the phosphate-specific transport system, it's a phosphate transport and sensing system that has a transporter in the inner membrane of the bacteria. When there's sufficient phosphate concentrations, the molecules will bind to key sensing proteins, which in turn inhibits transcription of what's called the faux operon. So this operon is important because during phosphate starvation, the repression is lifted and the transcription of the faux operon is activated. And this releases phosphate solubilizing factors into the environment. These would also solubilize the metals that are bound to the phosphate. So this is called the phosphate starvation response. So interestingly, here are the 4 hits that we had in our screen. Basically, what this means is that when we eliminate the sensing and the repression system, we actually get improved bioleaching, which makes sense because the bacteria basically think they're starved for phosphate and they're desperate to secrete things that will get them more phosphate. And also this would unlock more metals. So then taking a look at the strains that had reduced bioleaching capabilities, greatly reduced, what we find is that all of the really, really inhibited strains for bioleaching shared a similar mechanism as well. And this was not that surprising, but they were all associated with the mechanism for the oxidation of glucose into gluconic acid. So this includes the MGDH gene, which is the membrane-bound glucose dehydrogenase gene, which actually is the enzyme responsible for converting glucose into gluconic acid. It sits in the periplasm, the inner bacterial membrane, and it converts glucose into gluconic acid with the help of this cofactor PQQ. And so we also found some of these PQQ synthase genes, the genes responsible for making it, when they were knocked out, they also had greatly reduced biolixiviant production and bioleaching capabilities. So once we had honed in on these two key mechanisms contributing to acidification and bioleaching, we decided to engineer a microbe that would put them together for a really high efficiency bioleaching strain. We figured that we could eliminate the repression of acid production, just as in the knockout strain, we would delete basically the PST system. And then at the same time, we would turn up production of the organic acids by overexpressing the MGDH gene and some of its cofactor synthase genes to enhance the conversion of the glucose into acids. And to do this, also still in my postdoc, and actually this work is under revision, hopefully will be published soon. We tested overexpression of the MGDH with 3 different promoters. And we also explored knocking out a few of the different components of the PST system, but found that deleting the PSTS gene had the best effect on bioleaching efficiency without eliminating some growth defects that occur when you get rid of the PST system. So then we also put them together and did find a significant additive effect, especially under the control of one of the promoters overexpressing MGDH. So in general, we found very, very low pH of the biolixiviant, which correlated to a much improved rare earth bioleaching efficiency. So it was at this point I was wrapping up my postdoc, and I decided with my co-founder Sean Madim, we were going to actually try to translate this technology and commercialize it with REEgen. So we founded the company, applied for some grants, and began exploring what does it look like to scale this up, produce the biolixiviant in bioreactors, and leach rare earths from a variety of sources. And we were really excited to see that the genetic factors that we had modified and engineered this better strain weren't the limiting factors anymore. So once we started playing around with some process parameters, we were actually able to get the bioleaching with this new engineered strain to nearly 3 times more efficient for bioleaching over the wild type when directly compared side by side. So this type of efficiency has really yielded substantially improved economics for taking this into the field. So here's an example from coal ash where we leached the rare earths from coal ash, which can have high concentrations of rare earths from burning the coal, burning off the organic matters. We use media as a control, and then the wild type biolixiviant, the improved strain biolixiviant, concentrated gluconic acid and concentrated nitric acid. And in this case, and in many of the cases from many of the sources, we see almost as high efficiency using the S112 biolixiviant, this improved strain, as we do with the gluconic acid and nitric acid concentrated. So this is a really, really neat thing to find. And we've since, with these improvements, it's really tipped the scales on the economic model. So we've begun to focus more on, next steps, scaling this up, developing a continuous process, and starting to work with a bunch of different new materials to figure out where to really start this commercialization first. So I've given you an overview on the rare earth bioleaching side of our system. What the bioseparation side is not as far ahead. So Sean Madim, my co-founder, he was still a PhD student in the Barstow lab until the end of 2023. He just joined us in January. And I have to say that he's made incredible improvements with the separation since. So we do have some publications on this work, and we've also made more improvements since. So I'll walk you through some of that. So as an overview in bioseparations, the current method of deployment is that we're mobilizing these genetically tailored bugs for separating rare earth elements in hydrogel beads, and these beads are being packed into chromatography columns. We pass a solution of rare earths through the column, and the elements that bind more strongly, in this case, it would be the example showing NdPr, while those that they stay in the column are more retained, and then those that bind more weakly pass through more quickly, in this example, cerium lanthanum. So then the rare earth elements are then eluted with a weak acid, the eluent is reloaded to the column, and this is repeated until the desired purity is reached. So to date, we have demonstrated the functionality of these columns at REEgen, and we've also demonstrated their reusability several times with no loss in affinity. But to go back to some of Sean's PhD work, the key here is that we're using random mutagenesis actually for bioseparations. We're creating strains with higher rare earth binding overall, and also more selectivity for certain rare earth elements over others. So in his PhD, before he joined REEgen, Sean was able to improve overall rare earth bioabsorption to the microvibrio nitrogens with only a few rounds of random mutagenesis and only low throughput selection in 96 well plates. So as you can see here, he was able to increase overall binding by 48%. He also was able to demonstrate that this process yielded strains with better separation factors between rare earth elements, different rare earth elements, such as the better separation of the magnet relevant rare earths, your NdPr and dysprosium, from the much less valuable lanthanum. So it was really exciting to see that improving binding of a single rare earth also increased the separation factors. And we are continuing to push these separation factors higher today. So this work is published. You can find it in ACS Synthetic Biology. But now at REEgen, now that Sean's joined, we are supported by funding from the Air Force Research Lab, as well as the NSF through an SBIR award, to now translate this and start putting it into columns. So here's an example of one of his setups where he's partially packed a column with the beads to test the microbes to see, how many passes we can do and how pure we can get the rare earths with those multiple passes. Furthermore, we've also built a microfluidic device that will greatly speed up the selection process of the randomly mutagenized microbes, several orders of magnitude. This will allow us to rapidly generate and select new strains with greatly improved separation factors. So this is a silicone wafer with the mold for the microfluidic device that is used for this selection. And then the video is actually displaying a close-up of the cell sorting mechanism that helps us select the differential binding microbes. So moving a bit more into the company, so Sean and I, we founded REEgen in actually February of 2022, but we actually didn't start working I didn't start full-time until September in 2022. And this was really facilitated by getting an Activate Fellowship. So this comes along with a 100,000 grant, salary for me for 2 years, as well as travel and professional development stipend. So with this grant, it allowed us actually to license the technology from Cornell University. You're required to have some financial support before you can do this. And that licensing of the technology then also triggered our ability to enter the Praxis Center for Venture Development, which is our startup that supports us. It's on Cornell's campus. However, we remain an independent entity as REEgen. So since then, in only about 2 years, we've really been able to expand our team and push the research quickly because we want to develop this as quickly as possible because this problem is really happening right now. We also have a really great team of key advisors, including Marty Weems, who is no longer with Western Rare Earths, which is now American Rare Earths, but he was the CEO with Western Rare Earths and supported us throughout our research in Buzz's lab at Cornell and continues to support us now at REEgen. I mentioned that we've been able to raise a bunch of money that I'm incredibly grateful for. We get some funding through Activate, much support through Praxis Center, and also now we're part of the Venture for Climate Tech cohort this summer. And then we've also gotten grants from NSF, the New Energy New York, Fuse Hub Manufacturing Grant, and the Air Force Research Lab, as well as several pre-seed investors. In total, we've actually been able to raise dilutive and non-dilutive $1.5 million to date, which has been really, really incredible to get this going. So since we founded the company, we've been focusing really on business development and looking for our feedstocks until, as well as developing the technologies. So we've had over 200 different potential customer interviews. This has resulted actually in 15 different rare feedstocks that we've been testing all across the board. I didn't have minerals, but we're also testing minerals and trying to work with mining companies to see if we can help give them solutions for clean mining techniques. All of them have various values of the contained rare earths, the input costs, and their availability. And this has been a big part of what's informed our process and where we want to start, looking in order to recover these rare earths. So overall, we have actually through this process received 7 letters of support, and we have really high traction in particular among coal burning utility companies that produce coal ash and also smelters recycling other metals. And this is important because these smelters are producing slags that seem to have really concentrated rare earth from post-consumer waste. So they're recycling your steel, your platinum group metals, and a lot of these materials have rare earths, and those rare earths are going to the slag fraction and getting incredibly concentrated there. If we just think about the numbers, the US has 1.5 billion of tons of coal ash stockpile today. And we're still producing 100 million per year. However, this is a dwindling source. In contrast, the smelter slags, especially from recycled steel, they can contain from 100 to $500 per ton worth of rare earth elements, and we're still producing billions of these materials every year. So with that, we've decided to focus first on slag. And in particular, we're very eager to look at the slags from recycling platinum group metals because there's a high concentration of just recycled automotive catalysts. So these catalysts are typically stolen because they have platinum palladium. I think we all know that from the news, but they're actually part of this amazing circular economy for platinum group metals that they get taken in by large corporations that recycle out those metals and put them right back into AutoCAD. It's a beautiful story of circularity in the metal industry. However, there's actually higher amounts of rare earth elements in these catalysts than there are the precious metals. So they're not worth as much, but they're not getting recycled. In fact, globally, less than 1% of rare earths are being recycled today. So when we discovered this, we started working with some of these companies that are recycling AutoCAD to say, hey, where do the rare earths go? And they said, yeah, we'd love to know too, because we would love to stop having to buy them from scratch, sorry. And so that's how we started looking at the slags and did find that there actually is quite a high level of rare earth elements in both of these slags, in fact. In addition, both have the advantage that they have really decent levels of neodymium, especially the steel slag, likely because of those magnets coming out of post-consumer waste. And this is great because it's much higher than lanthanum and cerium, which can be major contaminants when you're trying to purify your NdPr. The cerium is pretty high on the PGM slag, but overall, the levels of rare earth elements are much higher, and you don't have as many of the other contaminating factors. So generally, yes, the PGM slag has much higher levels of rare earth elements than the steel, but they're both substantial, and plugging them into our techno-economic analysis, these are both serving as really great first sources for us to tackle. Also, excitingly, our clean solution, the biolixiviant generated by gluconobacter, the improved strain, can leach the rare earths from these slags when compared to concentrated nitric acid at 80 to 100% efficiency. It's a really great process and gets most of the rare earths out of it. So just to give you a sense of what we're working with when we talk about these PGM slags from AutoCAD recycling, they contain nearly 3% total rare earths, and about 1/10 of that is NdPr. This, to put into perspective, the highest total rare earth grade mine is the Mountain Pass mine in California. Their NdPr is also quite high, but it's interesting that the rare earth grade of the slag is actually comparable to many active mines around the world and much higher than several of the mines that we're trying to get going in order to help close the gap between supply and demand. So we at REEgen, we really feel this is a substantial, important source of getting, especially NdPr, for the renewable energy infrastructure that we will need in order to combat climate change. So this is a new source, a new feedstock, and you don't even have to dig it up. So on that, I will comment that the global rare earth market is really growing quickly, actually a double-digit CAGR, but much of this is really due to the demand for more neodymium to generate those magnets and renewable energy. This demand it's expected to increase to 2.5 times by 2050. However, our current mining capacity will only have the ability to supply about half of that. So there is definitely a huge need to find many more sources for this critical metal in order to get that renewable infrastructure for reversing climate change. So with that, I'll just comment on what our vision is. What we really hope to do is enable this renewable energy infrastructure. So to give you a sense, here we're based in upstate New York. If we could recover rare earths from the recycled steel slag, so this is slag coming from electric arc furnaces, we would be able to tap into a really expansive new source of neodymium, about 900,000 tons of slag per year. And we could supply enough NdPr from that slag to produce 240 wind turbines. I mean, we wouldn't produce it, but this is enabling that many turbines without having to mine more NdPr. And ultimately, this would offset 10.6 million tons of CO2 equivalent per year. So this is a huge part of our vision. While we do want to provide solutions for mining across the board, we really want to also tap into many new feedstocks for these critical elements. And so once at full commercial scale, we expect to be able to offset 0.5 gigaton of CO2 per year by enabling more of this renewable energy infrastructure, also the offsets from manufacturing rare earths cleanly. And then finally, a lot of our byproducts could replace Portland cement and hopefully avoid landfills completely. So with all that, we're pretty excited about where we've come only in 2 years, but we have a lot of work to do and we're trying to move as quickly as possible. So with the pre-seed funding available through Activate and the other grants and investing in dilutive sources, also our incubation at Praxis, we've been able to scale from those flasks and centrifuges that we were working with in the Barstow lab to prototyping our benchtop systems and separation columns. So our next step is actually to scale this to an integrated pilot system, and we hope to do that in the next year to 2 years. We're financing this currently through a raise that is currently underway. And then once we can prove out the revenue with a pilot system, we will deploy our commercial systems, and eventually we'll have multiple commercial systems on site with the slag to form rare earth concentrates. And then we will also have a rare earth bioseparations and manufacturing facility where we can then separate those concentrates to sell sustainable rare earth oxides. And that's about it. And with that, I want to thank you all for joining or watching this presentation. It's a pleasure to share this with you. And please reach out to me or my co-founder at any time if you have any questions or you just want to get involved or excited. I'm open to many collaborations. As you can see, we've got a lot of moving parts tech-wise. So we really think that this is a team effort and welcome ideas for how to move this forward more efficiently and quickly. Also, we are hiring, and please visit our LinkedIn page. The postings are there. Applications are through emails. Just read the directions and send us an email if you're interested in exploring a career with us. And yeah, thank you so much. I look forward to hearing from you. I'll take any questions.
And I want to say thank you so much, Dr. Alexa Schmitz, for a great, invigorating presentation. The work that you've done is amazing in such a short amount of time, and the opportunities that it brings globally is just really remarkable. And you made a comment about, your goal is that the trying to address some of the RE market needs for our growing economies and growing world, basically. And it was great to hear about all of the work from postdoc to current and where the plan going forward. What I'd like to do right now is open it up for question and answers. And it looks like we have one question queued up. Alexa, if you wouldn't mind reading that, and we'll keep moving.
Yeah, sounds good. So I have a question here from Yoshiko. Thank you, Yoshiko. So what type of treatment, if any, do you envision needing for the leachate prior to applying it to the bioseparation columns? And have you looked at recovering other non-rare earth metals from the leachates to improve the economics? Great questions. So I'll start with the first. And to do that, I'm going to take you back to our overview slide. It is true that this is leaving out some critical parts. It's not as very straightforward. It's much more complicated. So at the point where we leach the rare earth elements, there is a little bit of pretreatment that we do at this point, but we're exploring a variety of options. So the first option, which would be ideal, is that we could engineer a microbe that can quickly separate it as is straight off the agitator. So what we get in solution can go directly to a microbial column. And there's really no reason to believe this won't work, although We are also exploring some precipitation techniques to do some preliminary separation. It really does depend on the source. So in some cases, you have a lot of difficult contaminants. But the beauty of that is that we can also work with those leachates to engineer microbes that will tolerate the different inputs differently. So that's one of the beauties of our system. So yeah, some precipitation methods that we're exploring, but we're also looking at rare earth specific chelators, a variety of different chelators. This is part of a collaboration as well to see if we can just use a rare earth specific for all the rare earths to pull them out of the solution first, and then separate the different rare earths from each other with the separation columns. And then I think Yoshiko had a second part to her question, which was looking at other non-rare earth metals. And on that note, yes, there are other types of metals that are leaching. It has the leaching, I will say, has some specificity for rare earths, but other metals come into play. And many of them are very valuable. So definitely, it isn't a huge factor in our economic model because initially we can't afford to, well, can't afford. We could if we had the team and the money, but initially we're a small team, so we're really focused on getting those rare earths. But we are trying to connect with anybody externally who's able to process some of the other metals that we're getting. So if you're a company that is working with other metals to purify them, definitely this is open to collaboration to help us figure out what do we do with some of the maybe copper or zinc or vanadium that we're getting out of these materials. And eventually we're also applying for grants to maybe work on engineering the microbes in the separation columns to be specific for those other metals rather than just the rare earth elements. And at this point, we have some precedent that that should work. Thank you. Another question here from Dan. Great presentation, thank you. How important was the CMI funded work in your company's growth? So technically speaking, we haven't had any CMI funding for the work, but what I would say, maybe what you mean is the work that was done at Idaho National Lab. And I would say that was groundbreaking. That was really setting the fundamentals for what drove us to make this happen. So I'm still connected to them and look to them for advice at the company. And we're really hoping to continue working with the team that has done this initial work. So I would say Idaho National Lab's work was really fundamental to helping us get started. And they helped me throughout my postdoc as well. A question from Bo here. You said that part of the bioleaching efficiency increase was due to the phosphate levels. For your steel slags, do you find that the rare earths are normally phosphates or oxides? And if they are oxides, do they require pretreatment before bioleaching? Also, how are the reaction kinetics versus more traditional rare earth processing approaches? Okay, so in terms of the bioleaching efficiency increase being related to phosphate levels, this actually isn't the case because we've tricked the bacteria to thinking they're starved, but it's really not the case. Like we've made them hyperactively starving for phosphate, but it doesn't really matter at this point what the phosphate concentration is. So the efficiency is actually independent of phosphate levels. In terms of the steel slags, we aren't at this point, we don't know what the composition is, but I don't believe we're looking at phosphates for that case. We're pretty sure it's probably oxides. We still have to confirm this. No pretreatment before bioleaching. Well, okay, depends what you mean by that. We do grind them. So at this point we grind them, but like that's part of the techno-economic model. How much do you have to grind for this to work? And of course, the less grinding, the better, because that means lower energy inputs. Also, how are the reaction kinetics versus more traditional rare earth processing approaches? At this point, they're similar. I will say that we haven't really tested the traditional processing approaches directly. Like I said, we've been working with concentrated nitric acid, so it's tough to say that those kinetics are better. And this is something that we're working on right now, is putting together an experiment with a really, really comparable traditional rare earth approach to get those numbers. But great question, thank you. Question from Yongqing. I am curious whether the P limitation in the growth media in the bioreactor, with or without your genetic manipulation of the P metabolism, would be beneficial for pyrolytic production. This is a really great question, and We actually haven't tested it. It's a potential way of making this happen. However, it's only one part of the story, and we haven't tested it because the bacteria are so reliable in the way that we've engineered them that we don't really have to worry about subtle changes in media, especially when we're trying to set up a continuous process in the same tank, and those factors are going to change over time, most likely. So in the case where we would run into any issues with using genetically modified organisms, this would actually be a really great thing to test and see if it would help us at least somewhat. However, at this point, we do not actually mix the bacteria with any of the feedstocks that we're extracting from, so those microbes are contained and therefore not an environmental problem. So great question. Thank you, Cynthia, or Young Chin. Another question from Yoshiko. Since you are using live cells for the biosorption, do you envision any toxicity issues that would limit the reusability of the columns? Really good question, because we're actually not using live cells. They're live initially, but they don't have to remain alive. So what we find is that the columns, once they're encapsulating the microbes, they almost preserve them. And we're not testing to see if they're alive, but they shouldn't really be living in those situations. And we can reuse the columns over and over again. So and previous studies have tested if they're alive and they don't have to remain alive. So that's one of the really huge advantages of our system is we don't have to worry about toxicity issues with the columns. Thanks, Yoshiko. Question from Chong. After bioleaching, what is left in the residues, and are there environmentally friendly ways to deal with them? Also a really great question. So ultimately, we're hoping to be able to remove all of the harmful metals from the residues. Our vision is that we could even utilize the rare earth bioleaching to get the heavy metals in general. So I shouldn't even say rare earth, the bioleaching could be potentially a way to decontaminate solids, so especially when you have Superfund sites that have a lot of contamination that need to be cleaned up. We are wondering if this might be a really environmentally friendly way to extract the metals and then rinse them out of those solids. So instead of worrying about recovering and not diluting the metals, could we actually make really toxic soils with heavy metals cleaner and then also recover the metals for sale? Another question from Ali. Hi, Ali. I'm curious about the mixed organic acid biolixivian, as it sounds like gluconic acid is just one component. Very true. How important is the composition for the downstream separation? I don't have an answer to that question, but at this point, we are seeing that the chelation chemistry of the organic acids is affecting the downstream separation, and it's not making it necessarily harder. It's just changing what you'd expect, which is kind of cool because we can actually play with that a little bit to our advantage. At this point, we just have one type of gluconic acid, so we are doing sort of that treatment between the bioleaching and the separations at this point to accommodate that type of biolixiviant. But this is a great question, and we will be certainly testing this when we start changing the composition of our biolixiviant in order to leach the rare earths from different types of sources. Thanks. Another question from Matthew. In the development of a scaled up integrated process, what factors are you currently focusing on to optimize the separation efficiency of the bioseparations columns? What oxidation? I'll start with that one. So okay, so in terms of the separation efficiency in the bioseparation columns, we have your typical factors that have to be optimized. And to be honest, I am not as involved with the separation, so I don't know how well I can answer this question, but we're looking at flow rates and length of column, diameter of column, in terms of using this sort of bacterial resin for separating the rare earth elements. So these are standard column dynamics that you would need to optimize to do any sort of separation techniques. I hope that answers your question, and feel free to reach out and maybe talk to Sean, my CTO, if you wanted to explore more. What oxidation state are the rare earths in after the separation step? I don't know the answer to that question, and we haven't tested it yet. So great question, but I don't know. And what are the concentrations of the eluents roughly? Also, I'm not aware of that answer. at this time. But these are really great specific questions, and I would encourage you, if you really want to get into the nitty-gritty, please reach out to me and maybe find a time to talk with us. And if you're collaborating or looking for a position, I really would love to talk more and get you more involved. Awesome questions, everybody. I love answering and nerding out with all of you. It's been a real pleasure to talk about REEgen and yeah, share it with you. Any more
Great.
questions? Yeah.
We can answer another question if one comes up, but at this point in time, I want to thank you again for a great presentation. Also, great questions and answers. That was really just done very, very well. The recording, I want to remind our audience, and thank you, audience, for attending. I want to remind you that this is going to be available next week at aimslab.gov/CMI, also on our Critical Materials Innovation YouTube channel. Please join us again on September 24th when Thomas Tarka from the National Energy Technology Lab will discuss metallic, the minerals to materials supply chain research facility. Always information in Critical Times newsletter and also in our website. So if you have a suggestion again for a future webinar, something that you'd like to see, remember this is designed for CMI and by CMI. We'd love to hear about it. Make sure you put it on your exit survey or email us at cmi@mines.edu. And let's see, any other questions? I think we are good. Good to go. Thank you again.
Thanks, everyone. Thanks again for the invite. Take care.